Okay, everyone. Thank you all for tuning in. My name is Brad Weaver. I am a third year MD PhD student, beginning my PhD in Brad Cairns' lab up at the Huntsman Cancer Institute, where we study epigenetics, stem cells, and the concept of totipotency, or the cell's ability to contribute to any cell type of an embryo, including extra embryonic cell types. But that's not really the point of the presentation today. The presentation today is so that I can explain to you what I've been working on for my final project in this class, in Dr. Chapman's class. And this is a repetitive element parser. Basically, the idea here is that I wanted to design a script in Python that could take an output of a differential read count program that you typically get with an RNA-seq dataset, where you can compare two RNA-seq datasets and provide some important visualization functionality for sort of on-the-fly analysis and evaluation of your datasets. So with that background, what I'd like to do is build a little bit of background about repetitive elements. And I'm going to go through this fairly quickly just due to the time constraints. But these are actually really interesting elements and a big part of our mammalian genome, and I think that we're going to be learning a lot more about them in the future. So approximately half of the mammalian genome consists of these repetitive DNA elements, and they're long thought to be sort of junk DNA sequence and have no recognizable function. However, with modern sequencing technologies and improved molecular biotechnologies, we've discovered that up to about 5% of these transposable elements are involved in cancer development and gene expression in both good and bad ways. And importantly, they also provide important regulatory control for gene expression. And this sort of functionality is what I'm most interested in the early embryo. And we'll talk about that a little bit more when I get to my research. But by way of outlining the background here, let's talk about the general classes and more about their sort of what makes them unique. So there are sort of three main classes of transposable elements in the human genome: lines, long interspersed elements; signs, short interspersed elements; and DNA transposons, which basically encode or utilize a transposase enzyme to basically copy and paste themselves or cut and paste themselves at various points in the genome. So these have variable copy number in the human genome, anywhere from 20 to 40,000 for lines, over 1.5 million for signs, and 300-ish thousand for DNA transposons, and different fractions of the human genome are made up by these elements as well. For our purposes today, these class 1 endogenous retroviruses are a particularly special group near and dear to my heart that are derived from these autonomously transposing DNA transposons. And they're thought to arise by retroviral infection early in the human lineage, or the hominid lineage. Actually, the placental lineage, to be most correct, but the human lineage. And they sort of retrotranspose via this RNA intermediate, whereas the class 2 elements excise themselves completely and relocate to another area of the genome. That's not indicated here, but that's just sort of an important distinction. So if we look at how frequent these things are in non-coding sequence, we can see here, just like a general coding sequence of a gene, you have exons, which are the actual functional coding sequence of the gene that are going to be spliced together to make a protein eventually. We're at 1 through 14 here, and they're sort of located, interspersed throughout this DNA sequence. And all throughout, you have these different repetitive elements in the non-coding regions. You have ALUs, which are a type of line. You have signs, these other types of lines. You have LTRs, which we're going to talk a little bit more about in a minute, and SSRs. So it's readily apparent how abundant these DNA elements are, these repetitive DNA elements are, throughout the genome, even interspersed in important genes. So a typical LTR retrovirus is indicated down here, and this is what's thought to be a derivative of a retroviral infection that then integrated into our genome, and we've now co-opted this retrovirus, and now it's a quote-unquote endogenous retrovirus. And some of these retroviruses have intact reading frames that have coding sequences that still code for proteins. In this case, the three classic retroviral proteins are GAG, POL, and ENV, which are encoded here. And these LTR sequences are regulatory sequences that define the transcriptional and transposable abilities of these elements. And what's really interesting is that they're involved in the early embryo. And so in the Cairns lab, we're really interested in embryonic genome activation and how this embryonic genome activation relates to this concept of stem cell potency, and particularly totipotency, the ability of a stem cell to become anything, including a placenta or amniotic sac, depending on what kind of animal we're talking about, and how repetitive elements fit into this whole process and this whole logic. So basically, embryonic genome activation defines these first waves of transcription of RNA molecules in the fertilized zygote before the embryo implants in the uterine wall. During this cleavage stage development, the embryo undergoes massive restructuring and repackaging of DNA, going from a more packaged state in the sperm and the egg to a relatively less packaged state in the early embryo. And due to this, scientists who had been studying the transcripts of the early embryo saw this open DNA structure, but also saw that these expression of repetitive elements sort of coincided, mostly these endogenous retroviral elements. And they thought that this was just due to sort of how open the DNA was at this time, so you just sort of get expression of whatever. But recently, we've been discovering that these fluctuating endogenous retroviruses are critical for specifying lineage fate and stem cell potency at this stage. So our work in the Cairns lab is focused around understanding the process of EGA, and using that knowledge to contribute to our understanding of pre-implantation development, as well as thinking about how to leverage these discoveries for improving the efficiency and functionality of stem cell-based technologies in regenerative medicine. So here's a diagram of just tying all these things together. And in the upper left, we have early pre-implantation development, where you have gametes fusing together to form an embryo that then divides into a 2-cell stage embryo, which is when embryonic genome activation, EGA, occurs. And these are the particular transcripts that are abundant at this time. Importantly, this murine endogenous retrovirus type L is super abundant. And then that sort of dies off before you become a morula and a blastocyst. And if we look at the structure of this MERV-L, here's the internal coding sequence. It still codes for an open reading frame of a GAG and a POL. And it has these LTR sequences that are further subdivided based on sequence, which isn't really important for us right now, into MT2 and MT2M. And then here's just an electron micrograph, and here's a immunofluorescence image showing that you can tag these viral particles with antibodies and see that they're abundantly expressed at this 2-cell-like stage. And then that sort of goes away by the 8-cell stage. In the germinal vesicle of the egg, it's non-existent. Here's electron micrograph showing a similar thing. So in the early reprogramming of the embryonic genome, this coincides with the transcriptional burst and MERV-L reactivation, in the mouse at least. And in the human, actually, there's a subset of endogenous retroviruses type L called human endogenous retroviruses type L that are similarly expressed. So we find this really interesting, and what I wanted to design was a repetitive element parser that could allow us to sort of on the fly take these RNA sequencing data sets and determine what, if any, important repetitive elements are being expressed and how much they're being differentially expressed. So for the rest of the presentation, I've got a couple minutes. I want to take you through why this is important. So basically, you have a few dependencies that will allow us to run this package. NumPy is important, Pandas is important, IPy widgets functionality is going to be coming later, and then to append this data set to a database, I'm using SQLite Alchemy or SQL Alchemy. So the first step is going to be taking this Excel file that's the output of a differential read count program, and here's sort of what the Excel file looks like. You have genes, the families of those genes, where they're located in the genome and their name, the exon length, etc., etc. What we really care about is sort of the base mean expression and log two-fold change over control. And we're going to see why those are important in just a second. But basically, we want to take this big, nasty data set and extract only the meaningful information that we care about. So we're going to import these dependencies, and then we're going to take this gene list we're going to set these variables as our pandas data frame variables and read in these Excel files. And these are two separate sheets of the Excel file, just reading into different pandas data frames. So we're going to mostly focus on gene list 2 for simplicity right now. And once that gets imported, I'll take you through this class that I've defined below called a differential expression data frame class. Basically, this init method takes one argument, which is the data frame that we pass to it, which is going to be this gene list 2, and it's going to define a new data frame, fill in any NA values, and then append a new column to that, which is a base mean log 10 expression rather than just the base mean expression. And that'll just help us with visualization a little bit later on. I'm going to append this new data frame to a SQLite database using this pandas data frame to SQL functionality. And then from there, I want to slice it up, and this is really where the flexibility of this program comes in. I want to slice it up into IRV-L family, lines, signs, and LTRs just for our visualization purposes. And we can do this all using pandas syntax right here and then visualize it from there. The string method just returns an explanation of what the object can do and what functionality is possible. The most important functionality possible at this time is the MA plot, which takes a single argument, which is defaulted to all, which will determine what we're going to visualize on the back end of this. So all means we're going to visualize both LTRs, IRVs, lines, and signs. And then beyond that, the user can also specify just LTRs, just IRV-Ls, lines, or signs. And if they type in signs or anything else, it's just going to give them back signs. And then these are the bokeh documentations for creating these important scatter plots. And we got a little bit of tooltips functionality, and I'll explain why that's important in just a little minute. So let's run that cell, make sure everything looks good. What we're going to do is create an instance of this class object by passing this pandas data frame, this gene list 2 that we defined earlier, into the object as an argument, or into the class as an argument, excuse me, and creating a variable instance like that. So let's just run that. And then what we're going to do from there is use this MA plot method and just call it with nothing in it, which should return all by default. I'm just going to call that. So what that does is it prints out a new HTML web page, which shows us the LTR expression, IRV-L expression, line expression, and sign expression, which is nice. And then we can hover over these individual points of interest and get the names of the particular LTRs, LTR 7Y, MER 66B. So that can sort of tell us what's important in our data and what's not. We have MLT2A1 over here under IRV-Ls. Under lines, we have the most expressed being L1P4B. And in that way, you can sort of get a sense of the distribution of your data and how much it has changed, and also the important single points of your data. And so I think this will be really helpful moving forward for us in our analysis pipeline of some of these repetitive elements. Furthermore, you can just plot this inline, and not sure why that's not working, but it's not super important right now. So either way, from there I went on and defined this IRV-L subclass that inherits functionality from the parent class of differential expression data frame object. And basically here, what I wanted to be able to do was identify some important IRV-Ls based upon log fold change over control. And we're going to set this threshold to be 3. Basically, this is designed to return part of the IRV-L family that has a log fold change of greater than or equal to 3. And so basically I can pass in this this gene list again, and I can set my variable to IRVs and call the instance of the class of IRV-L by passing in this gene list, and ask what are the important IRVs by calling the method IRV-L up. And I can use the method call IRV-L up to ask what the important ones are. It didn't work just a second ago because I didn't run this cell, but now it has run, and it has spit out MLT2A1 and IRV-L internal which have base mean, which have log 2 fold change of 3.24 and 3.009 respectively. And so we can see that that's consistent with our data before, where we have ERV-L expression right here, and the two most highly expressed are MLT2A1 and HERV-L internal, and they're both above 3, as we can see right there. So I really think this repetitive sequence analysis pipeline will help me and my colleagues in the lab just assess sort of on the fly what important points exist in our data, and how we might ask other questions based upon the particular data points and also the spread of the data. So with that, further functionality that I'd like to include in the future is a command line interface. So this can be more broadly applicable to people who don't know Python. And that's something that I'm going to be working on in the near future. So thank you very much for your attention. I really appreciate it. Thank you.